Hello and welcome to part one of chapter 8. This is the chapter on blood and clotting. This is a general overview of what we're going to cover in this lecture. First, we'll hit the normal blood components, what's supposed to be in blood, and then we'll get into the disorders that we see in blood. The two most common problems are referred to as anemia, not having enough red blood cells, or not having enough oxygen carrying capacity. And we'll cover that in part one. And then in part two, we'll get into the clotting disorders. The two main problems there are clotting too much, hypercoagulation, or not clotting enough, hypocoagulation. But that'll be in part two. This is a big laundry list of disorders. Again, these are fair game for the patho blog if you haven't done yours yet. The ones that have asterisks next to them are things that we're going to touch on, at least touch on in these two lectures. All right, so normal blood components. There's the plasma, the liquid part of blood, and then there's the more solid bits, the formed elements. In the plasma, it's primarily water. We also have our electrolytes dissolved in there, sodium, potassium, calcium, magnesium, etc. Small molecules, the two most important, the two that we associate with the respiratory system, oxygen and carbon dioxide. And then there's the nutrients, glucose, amino acids for building proteins, fatty acids related to lipid metabolism, and other molecules related to this. Larger molecules are big. These are the proteins. And you'll remember complement protein system and the clotting protein system. We've mentioned those in passing before. These are proteins largely made by the liver. The one we haven't mentioned before is albumin. The vast majority of the protein that's in the blood is actually albumin. And its biggest role is that in water balance. I think you're all probably familiar with the term osmotic pressure and having water shifting because of osmosis, because of things like the presence of sodium ions. A similar thing happens related to the proteins. It's referred to as oncotic pressure. It's a similar concept, though. It's wherever these proteins are, that's where the water is going to follow. And so a big part of keeping water in our circulatory system is related to the presence of albumin. And for example, when I think we mentioned this in the immune system chapter, if the liver is malfunctioning because somebody is suffering from cirrhosis or has liver cancer, there's a decrease in the complement proteins that are made, but there's likewise a decrease in the clotting proteins and in the albumin produced. So we're not retaining the water in the cardiovascular system, and so that's where the puffiness comes from. A lot of the water that kind of leaks out into the tissues because the albumin's not holding on to that water anymore because there's not enough albumin. Lipoproteins, so related to moving lipids around. Lipids, they're not going to dissolve in water, and so they have to be bound to a protein in order to get it through. This is for moving cholesterol around, for moving other lipids around. These are the lipoproteins, also liver dependent. And then we get to the so-called formed elements. The vast majority of those are red blood cells, erythrocytes. And we will be talking, so the function of the erythrocytes is primarily to carry oxygen. There's also some carbon dioxide shuttling that happens. White blood cells we talked about in great detail in the immune system, so we're not going to go over them again in this chapter. And then platelets we have not mentioned too much. Platelets are fragments of cells and that are involved in clotting, and we'll get to that later in the chapter. This material covered on page 241. All right, let's talk about some lab values that come up. The things that are highlighted in yellow are the things that you're probably going to use the most. The one with the asterisk, this is the most important one, the hemoglobin level. This is the amount of this hemoglobin protein in a given volume of blood. this is how diagnoses of anemias are made. You'll often hear people talk about H&H, or hemoglobin, and hematocrit. Hematocrit is the older measurement, and I'm going to try to describe it over here. This one is still used in certain contexts. It's still actually measured, but in most lab reports, it's still given, because some of the more traditional providers still use hematocrit. They think in these terms, in terms of these percentages, but for the most part, in lab printouts that include a hemoglobin level, the hematocrit isn't even measured. It's just calculated based off of the hemoglobin. Based on the mean cell volume and reticulocyte. Good heavens, I'm having trouble. Reticulocyte count. So let me tell you what the hematocrit is. This is when we take a volume of blood. This is often done from a finger prick and use a capillary tube. I know that this is just showing a test tube, but you use a capillary tube. It's a thin strand of glass with an opening in the middle. And due to capillary action, the blood is drawn up this capillary tube. Then that capillary tube is spun down in a special centrifuge that, it only takes those very, very thin capillary tubes. So the components of the blood are separated. The red blood cells drop to the bottom, and then there's that buffy coat, and then there's plasma. And the capillary tubes are marked so that you're going to get about 10 centimeters of blood. And so then you can easily measure. This is, say, 10 centimeters long. And then you see how much of those 10 centimeters are red. And if that's 4.5 centimeters, then we say that hematocrit is 45%. And that's how you do a hematocrit. Where you would do something like this, for example, if you're out in a clinic that's far away from its own access to a lab. For example, when I was working up in Hatch, we had a clinic, we had a lab in the clinic, but we didn't have access to hemoglobin levels. So we would do a hematocrit so we could get that value right away and help the provider, help the patients. So you as providers may need to do a hematocrit measure, but for the most part, you're going to work off of hemoglobin numbers. I'm not going to make you memorize these values. They're always given on lab reports. They'll also deal with mean cell volume, MCV. Sometimes you'll see this called mean corpuscular volume. It means the same thing. How big are the red blood cells? And why that matters is it helps you to diagnose different kinds of anemias. And the reticulocyte count. These are not quite mature red blood cells. So it gives us an indication of how well is the bone marrow replacing the red blood cells. All right, red blood cell production. Erythropoietin, EPO, this is the hormone that is released mostly from the kidneys that signals the bone marrow to make more red blood cells. You'll remember the line of cells that gives rise to most of the white blood cells also gives rise to the erythrocytes. And so the erythropoietin signal is the one that says, make me more of these red ones. That's what we need right now. Now, for making the red ones, we have high requirements for iron, for making the hemoglobin, as we'll cover shortly. And then these two other nutrients come into play, folate and vitamin B12. You may know folate is also known as vitamin B9, not super important, but it's a B vitamin. And both of these are required for the DNA synthesis when the cells are first dividing and becoming the cells that give rise to the erythrocytes. And I'll cover that in more detail later. Anemias can result from deficiencies of any of these items above. Don't have enough EPO, not enough iron, not enough folate or vitamin B12. All of those can result in not having enough red blood cells. Let me go over this term. I love it. Emias, you'll know, means in the blood. So if we have bacteremia, we have an infection in the blood, or hyperglycemia, it's too much sugar in the blood. Anemia, AN, means not. So you don't have enough blood in the blood. So not having enough red blood cells in the blood, or not having enough oxygen carrying capacity in the blood. Let's talk a little bit about hemoglobin. The normal hemoglobin we use is hemoglobin A. A stands for adult. This is in contrast to hemoglobin F, which stands for fetal. The hemoglobin you find in a fetus is slightly different from the hemoglobin that you find after birth in everybody else. Let's see, it's a protein, largely a protein. It has 4 protein chains. 2 of them are referred to as alpha and 2 of them are referred to as beta, but 4 protein chains. Each one of them holds something called a heme group, represented in our textbook by these black structures. This is a different cartoon from a different source. I like the way it simplifies the structure of the protein so that you can actually see the heme groups in here. And the little ball in the middle represents the iron. And each of the irons can bind an oxygen molecule, an O2 molecule. So making the hemoglobin, the limiting factor is typically the iron availability. You can usually make the protein part. It's that iron in the middle that sometimes is in short supply. So during iron deficiency, the hemoglobin synthesis rate decreases. So we can't make hemoglobin fast enough, and we can't make the cells fast enough. And the cells we make tend to be small. All right, so here's the hemoglobin. I mentioned that there's these 4 chains, and 2 of them are alpha chains and 2 of them are beta chains. The thing that we're going to be showing here, and we'll look at in more detail shortly, is this beta chain. And we'll get into that for sickle cell anemia. What I wanted to reinforce here is hemoglobin is a protein. And like all proteins, it's made of amino acids that occur in a particular order. And so the order of those amino acids, you may recall, that's referred to as primary structure. And then once you have this long chain, it's hundreds of amino acids long, then that thing gets folded up and becomes this 3-dimensional structure. When it works right, you have a whole bunch of alpha and beta chains put together. These are the full hemoglobin molecules. And these things kind of float past each other. If you can picture balls in a ball pit, they're touching each other, but they're not actually attached to each other. They're just passing by. And they're floating around inside this famous biconcave disc, the red blood cell. This stuff, again, is covered on pages 244 and 245. And then it's the heme groups here in the center that each have an iron molecule, each have an iron atom. And so they can each hold an oxygen molecule. All right, so some genetic abnormalities of hemoglobin. The buff colored background indicates we're switching topics a little bit. We're going to cover 2 main genetic abnormalities. The first one is sickle cell disease, sickle cell trait. We're going to do a little genetics review. This is an autosomal recessive disorder. Autosomal means that it occurs somewhere other than the X or the Y chromosome. I'm not going to expect you to know which chromosome it occurs on, just that it's autosomal, meaning you're going to see it equally frequently in males and females. It's a single gene defect. It's the code for 1 protein that is messed up. A little review of how genetics happens. Here we have 2 parents. They're both heterozygous. They're both carriers. So the normal copy, since this is a recessive disorder, the dominant condition is the normal one. That's represented by capital letters. So that's the normal copy of the gene is capital A, and then the mutated copy of the sickle cell disease gene is little a. If you have 2 heterozygous parents, there are 4 different possible combinations that they can make, and their offspring will come out as. If they make the homozygous dominant, capital A, that kid is completely unaffected. These other 2 combinations, they're both heterozygous, and really these are indistinguishable from each other, whether they got it from mom or whether they got it from dad. These are both referred to as carriers. With sickle cell, if you have 1 copy of the mutated allele, you may have some signs, but generally this doesn't rise to the level of a disease, of a clinically significant problem. It's in this condition where the child inherits both a little a from mom and a little a from dad, and they're homozygous recessive. This individual would have sickle cell disease. This is covered on these pages. All right, so what is sickle cell disease? What's the problem? We say that somebody who has that sickle allele, they make something called hemoglobin S in contrast to hemoglobin A. It's a single base pair mutation. There's 1 typo in the DNA, and it's in a very specific spot, and it leads to 1 very specific mistake. In that chain of amino acids, it's the one in the 6th position that gets substituted. It's instead of a glutamic acid, a valine comes in. These are just 2 different amino acids. Glutamic acid is a hydrophilic one. It mixes well with water. Valine is a hydrophobic one. It does not mix well with water. And that has huge consequences, as we will see. So it's in the 6th position here, this hemoglobin S. You've already seen this diagram. We've got the normal primary structure. This is in the beta subunit. It folds a certain way and becomes part of this larger hemoglobin molecule. And these just float around each other here in the biconcave disc. In the case of hemoglobin S, there's a mistake in the DNA that leads to this primary structure, such that this amino acid in the 6th position is replaced with this amino acid in the 6th position. Why that matters is that this beta subunit is shaped a little bit differently, and it has this section that's a little bit hydrophobic. It doesn't dissolve well with water. The hydrophobic things tend to find each other. When they're in solution, they're more comfortable avoiding water, so they're more comfortable with each other. And so this has huge consequences for the structure of the red blood cell. So here's the sickle cell hemoglobin. That beta hemoglobin it's just a little funky. What the consequence is under certain stressed conditions, like under hypoxic conditions, this changes the shape of this molecule so that they're not floating by each other like little balls just floating by each other in a big ball pit. They get changed in shape so they're more like Legos. So they pass each other, but then there's a part that can click and they can kind of connect to each other. And when that happens, instead of just having this big ball pit, what we have, we make these long fibers of the Legos, and they just longer and longer and longer. And it's a little bit rigid, and it changes the shape of the overall cell. So where you had this very soft and malleable cell that could fit easily through tiny capillaries, it just changes its shape a little bit and it just squeezes right on through. Not anymore. Now we have these semi-rigid rods running through the cell. So it causes this to form this kind of sickle shape. And that stuff gets stuck in the little capillaries. It's supposed to bend and go around the corner, and this cannot bend. And I'm going to give you a way of thinking about this. It's not perfect, but do you know what an underwire bra is? All right, so the top one, this is like a no wire bra. It's very flexible, things can move easily. This would be like, it's a little bit rigid. It's not solid completely, but it has a little more form to it. And so it can't fit through some of the tinier spaces. And it's just a little more rigid, which is enough to allow the forces in circulation to tear the cell open. And this is how some of the sickled cells get damaged and they get taken out by the immune system. This is our textbook's representation of the same principle. It's on pages 245 and 246. Here's the hemoglobin A. This is like the balls in the ball pit. Hemoglobin S has these bits on it that are a little bit hydrophobic. And then under hypoxic conditions, they're a little stressed, the molecule changes even more, making it more Lego-like so that they can click onto each other and you make these long strands. And under the worsening conditions, you get more and more growth of these long strands. So dehydration will do it, hypoxia, significant drop in temperature. That is going like in the desert Southwest in the summertime, going from very, very hot conditions to coming indoors to air-conditioned condition, drop the temperature. In the winter, it's the opposite. You're from a warm house and you go out into the cold, freezing, and temperature drops again. What's happening is the blood vessels are vasoconstricting, and it's enough to trigger this. So the growth of the hemoglobin S polymers, the red blood cell gets deformed into that kind of semi-rigid crescent shape, and sickle cell crisis is precipitated. The thing is that makes the situation worse because we've got an area where these little sickled cells are blocking blood flow. Well, that's going to make hypoxia even worse. And then, so this gets worse and worse and worse. And as you know, hypoxia hurts. This is a painful condition for our patients. I think that's it on sickle cell. We're going to move on to a second genetic disorder. I'm not going to talk about it for long. In the thalassemias, so it's a genetic condition. Different versions are all recessive in nature. These are mutations of the hemoglobin beta chain or the hemoglobin alpha chain. It produces what's known as the thalassemias. In both of them, the red blood cells are microcytic. They're small, hypochromic, they're pale due to the reduced hemoglobin concentrations. These are treatable. The patient sometimes needs transfusions, but if you are aware that the patient has this problem, it can be treated. We don't have a fix for it yet. And the same with hemoglobin S. There are genetic therapies that are being attempted. And there's one, if you're interested in it, put a message in one of the discussion boards or the discussion board for module 2, and I'll address it a little bit more. All right, so those are the two genetic conditions leading to anemias that we're going to talk about. Now, buff colored background, meaning we're switching topics. How do we diagnose anemias? Primarily, we're working off of hemoglobin levels. Low hemoglobin levels tells us. I included hematocrit here because you will still hear people talk about hemoglobin and hematocrit. But as you know, unless you're in a lab that doesn't have access to the hemoglobin number, the hematocrit is just a calculated number. So the low hemoglobin gets the diagnosis of anemia. And then if you look more closely into the evaluation of the blood smear, this gives you hints as to what the cause may be. So looking at the size of the cells, if they're microcytic, if they're small, that may give us certain indications, as I'll talk about shortly. If they're normocytic, they're the regular size, then the anemia may be resulting from something like we're losing the blood. The cells are being made fine, but then they're going away somehow. Macrocytic, this is counterintuitive, I understand, but you can have low hemoglobin levels and big cells, and there are some very specific dietary deficiencies that can lead to that condition. And so these clues help you to figure out how are you going to treat this patient. The other thing that's noted is how dark is the cell? What does it look like under a microscope? And so hypochromic, it means not enough color. The color comes from the hemoglobin. That's the red color. And so if they don't have a lot, it indicates low hemoglobin concentration. And then again, altered shapes. This gives you indication of what the problem may be. Covered on this page. All right, some general concepts about anemia. What we find clinically depends on what is it that's leading to this problem and how long has the problem been going on. If the problem is acute blood loss, hemorrhagic anemia, somebody lost a bunch of blood because maybe they just delivered a baby or they were injured severely, they're losing a lot of blood, the patient may be hypotensive, if they may be going into shock if the blood loss is severe. Some of the other things that go along with this is the racing heartbeat, the fast breathing. They're not getting enough oxygen to their tissues because they don't have enough red blood cells to deliver the oxygen, and they may pass out. If the problem is a chronic rather than an acute problem, the kidneys kind of have a chance to help replace the blood volume, and they compensate by holding on to sodium and water. How they know to do that is as the blood pressure goes down, the body starts releasing, we have aldosterone as part of the renin-angiotensin aldosterone system. So aldosterone causes the kidneys to hold on to sodium, which causes the water to follow the sodium and keep the blood volume up that way. We can also release antidiuretic hormone, which causes the kidneys to hold on to just water. So that's going to expand the plasma volume. The patient may still be tachycardic and tachypnic because they're still not getting enough oxygen, even though the blood volume is still within a normal range. So the sense of fatigue and exercise intolerance, that's not going to be as dramatic as what happens with acute blood loss. It's going to come on more slowly. It may take the patient a while to identify that there's a problem. Now, as I hinted, there are some anemias due to dietary deficiencies, and we're going to do those next. Iron deficiency anemia, by a long shot, this is the more common ones. This is the most common anemia worldwide. And let me remind you, here's a giant hemoglobin. It's got those 4 chains on it. Each of the chains is holding a heme group. That's represented here. This is this molecule. It's a heme group. At the center of the molecule, there is an iron atom. So each of these hemoglobins has 4 iron atoms. And then each of the iron atoms can hold an oxygen. And you've probably had this thought before that it's just the iron that's missing. I can still make the heme group. I can still make this big hemoglobin. What is the big problem? And this is one way of looking at it, is that the iron is the part that actually binds with the oxygen. So even though the rest of the molecule is there, the molecule cannot do its job of binding the oxygen. And the way to think about this is, if you can think of a performing artist that you know and like, and let's say you were planning to attend a concert. And turns out that the performing artists themselves, they can't make it to the concert, but the backup band is still going to be there, and all the backup dancers and the lighting guys are still going to be there, and the ticket takers are still going to be there. All the other stuff is going to be there. It's just that one person who's going to be missing. I mean, would you still go to that concert? And most likely the answer is no, even though all the backup stuff is still there. This is why we're going. And same with the oxygen. If the iron's not there, the oxygen is not going to bind. And so even though you can still build the hemoglobin, it's not going to do you any good. It's not going to do the patient any good. All right, so why are we missing iron? It could be because we had the right amount of iron previously, but all of a sudden there's this huge increase in requirement. That happens during pregnancy. There's a huge expansion of the blood volume. And in early childhood, toddler time, infancy and toddler years, You'll see some iron deficiency anemias at that time, and in adolescence, when kids go through the second giant growth spurt that they go through in adolescence, we see iron deficiency anemias. It could be because of chronic blood loss. Somebody may have very heavy periods and they're losing significant amount of blood. I know it doesn't seem, I mean, it's not buckets and buckets, but still, this is all, we recycle our hemoglobin. Well, we recycle the iron from the hemoglobin. And if instead of recycling these old red blood cells, we're just losing them, for female patients during the menstrual years, we may see iron deficiency anemias. Gastrointestinal bleeds. It seemed like, what is so wrong with the gastrointestinal system that's always leaking? Well, think of it this way. Every other system can also leak blood, but in every other system, the patient's going to notice. It's not going to go undetected, either because they're leaking into a cavity, then the pressure is going to build up, or because it's actually leaving the body in the form of urine. You're going to see the red. But think about what else leaves the body regularly and is dark enough that you're not going to see the blood there. And that's in the GI tract. Unfortunately, often these chronic GI bleeds are associated with the development of a cancer. It could be due to an ulcer, but it's often cancer related. And then decreased GI tract iron uptake, either because they're not getting enough iron in the diet or they're not absorbing it well enough. All right, we mentioned that happens in infants, resolved with iron-containing foods like some commercial formulas. They're iron fortified, I think is the term they use. And we also see iron deficiency anemias in older adults, though you do have to differentiate from other anemias. Indicators of iron deficiency. The mean cell volume is going to be small. This is microcytic. They're going to be pale, hypochromic. But here are the more specific tests that are done to identify if this is an iron problem. So iron does not travel as loose iron in the bloodstream. It can't. It's damaging to our cells. And so it's bound to some other proteins. And here are the two most important ones. Ferritin, it can hold on to hundreds of iron atoms. Most of the ferritin is tucked away in the liver. And humans are not the only animals that do this. This is why people who are low in iron are sometimes encouraged to eat liver, not human liver, but liver, because there's lots of ferritin with the iron in liver. However, some ferritin kind of leaks out into the plasma. And so now we know you don't have to biopsy the liver to get a sense of how much iron is in storage. You can just look for some of this ferritin that's out in the plasma. If that ferritin level is low, that tells you that the person's long-term storage of ferritin is low. They have low iron stores for the long term. The other protein to look for is transferrin, movement or cross. So this is for ferrying the iron in the blood. This one, transferrin, only holds 2 iron atoms on it. And so here's the weird thing. The transferrin number actually goes up because the bone marrow, well, where's my iron? I need iron to make the hemoglobin so I can make you some red blood cells. So the transferrin number goes up, but if you look at transferrin saturation, how much of this transferrin is actually carrying iron, that number is down. So with iron deficiency anemia, it's been going on for a long time, the ferritin, the long-term storage is down, and the transferrin saturation is down. But the amount of transferrin, the indication that you're trying to transfer iron, that shows that the body's trying to move some iron around. This is explained on these pages. All right, now a couple of other deficiencies that can also lead to anemias. They're both B vitamins, vitamin B12 and folate. yeah, folate is B9. A way to remember that, everybody remembers folic acid for pregnancy, B9, 9 months. That's not why it's B9, but that's one way of holding on to that information. All right, so B12, vitamin B12 is also known as cobalamin. I don't know how to remember that other than it's cobalamin. Both of these, B12 and folate, they're both required for making DNA. And I'll tell you how that comes into play. The deficiency of either nutrient slows down the development in the replicating cell. Now, this is where it's counterintuitive. It actually leads to bigger red blood cells. So instead of microcytic anemia, like in iron deficiency, you get macrocytic anemia. And I'll explain on the next slide why. This is where deficiencies can come from. One thing is vitamin B12, it's only found in animal-derived foods. Unlike pretty much every other nutrient, we always talk about fruits and vegetables. You can eat all the fruits and vegetables you want. B12 is not going to be in them. So this comes from meat, eggs, dairy. So the folks who suffer from these kind of deficiencies, generally vegans, people who are really strict about not eating any animal products. Now, people who are that strict about their dietary intake tend to be pretty careful about their dietary intake, and they're aware of this problem and they generally supplement already. But if you have a patient who's not on top of their diet, they just in general are avoiding animal products, they may be low in B12. With folate, this follows the general pattern of nutrients in fruits and vegetables. I remember this folate foliage that's in leafy things. So folks on highly processed diets, not a lot of fruits and vegetables, they may be deficient in folate. And then we're going to go into more detail about this larger than normal red blood cells on the next slide. All right, so this is normal red blood cell development. We're in the bone marrow, we've got the stem cell. The cell undergoes mitosis, divides into 2 cells, and then those cells are in G1 and they're growing. And you know all the signaling that has to happen to say, okay, now move on into synthesis. So copy the DNA and then move into G2. And then now we're ready to divide. And now we've got 2 cells again. And we have normal sized red blood cells. In the case of B12 or folate deficiencies, it's a similar process. You start out with the same stem cell, and the cell's growing, and we're in G1, and we get big enough, and say, okay, move on to synthesis, start copying the DNA like normal. But man, that step has taken a long time because we're having trouble making the nucleotide. We're having trouble making the A's, T's, G's, and C's. Well, I guess while we're here, we keep making hemoglobin. So that cell keeps getting bigger and bigger and bigger. But we're stuck in synthesis for a really long time. And by the time the cell's finally done copying the DNA and it goes to divide, you have a much bigger cell. So you're making fewer cells, but by the time you get them to be mature red blood cells, they're bigger than normal. That's where the macrocytic part comes from. All right, and now we're going to zoom in a little bit more on specifically B12 deficiencies, because there's some things about B12 that are a little different from folate and can lead to this problem. All right, so the B12 absorption is a little more complicated than the folate absorption. Absorption of B12 requires the presence of this other thing. It's called intrinsic factor. It's an old name, we're stuck with it. It's made in the stomach. There are some gastric parietal cells that make this stuff called intrinsic factor. The intrinsic factor has to find the B12 and they actually bind to each other. And it's that combination, that fused thing, the B12 intrinsic factor combo, that gets absorbed in the small intestine. and the tail end of the small intestine specifically. So if there's a disruption to the gastric parietal cells, even if the person's getting plenty of vitamin B12 in their diet, they're not going to be able to absorb the B12 because it has to be bound to the intrinsic factor in order to pull it into the bloodstream. So who's going to have this problem? Well, there is an increasingly more common bariatric surgery called gastrectomy. It's making the big pouch of the stomach, or making the big stomach into more of a sleeve. So a lot of the real estate of the stomach is removed. Or the gastrectomy can happen for other reasons because there's a disease in the stomach, and so you have to take off this piece. This one's less common, but also happens. There may be autoimmune destruction of specifically these parietal cells. The immune system starts attacking those cells, and this can lead to a condition called pernicious anemia. Pernicious means deadly, again, old-fashioned term. You'll sometimes hear the term pernicious anemia applied to any vitamin B12 deficiency, but technically pernicious anemia refers specifically to the B12 deficiency that's due to this autoimmune problem. All right, for whatever reason, we don't have enough intrinsic factor being made. That's going to lead to a B12 deficiency because we can't absorb it well. As we've mentioned before, this anemia is going to be macrocytic. The cells are going to be big. Another thing that goes along with a B12 deficiency that you will not see with a folate deficiency is it's also required for the myelin formation. Those are the sheaths around the axons. So the patient may have neurological deficits that go along with this, especially sensory paresthesias, tingling, numbness. If we supplement B12, the anemia will generally reverse, but some of the neurological problems may be irreversible because that damage may be permanent. How to supplement. So for some patients, for example, bariatric surgery patients, some bariatric surgery patients, they just need to supplement with oral B12. Others, because they don't have enough of this intrinsic factor, so you can supplement all you want, but they're not going to be able to absorb it. And especially somebody with the autoimmune problem, they're going to need to get that B12 directly into the bloodstream. And you can do it in a couple of different ways. There's sublingual things. So there are some blood vessels under the tongue that can absorb, the B12 can be absorbed through the tissue and get into the bloodstream that way because you're bypassing the digestive system for that. The other way is just to do injections. It could be weekly, they could be monthly, depending on how deficient the patient is. The injection is cyanocobalamin. Doesn't matter. Moving on. Just the patient needs to get some B12 in their system. Okay, now we're moving on into miscellaneous anemias, and then we'll wrap up this section. There's anemia of chronic inflammation. It can be due to chronic infections, autoimmune disorders. Without going into a huge amount of detail, there's iron in the body. So the ferritin value will be normal, but it cannot get to the bone marrow. So the patient has enough iron, but not in the right place. So the cells that are going to be made are going to be microcytic, as if they had a real complete iron deficiency. The low reticulocyte count, they're not going to be making a lot of little red blood cells because they don't have the supplies. So here's the paradox. Ferritin levels are normal, but they're still, it's a kind of iron deficiency. And the best management is to manage the underlying chronic disease to help reverse this problem. Anemia of chronic kidney disease. You'll remember erythropoietin, EPO. This is the signal to the bone marrow to make red blood cells. That comes from the kidney. I know it's a weird combination, but that's where it comes from primarily. There's a little coming from the liver. So patients with kidney disease so the amount of EPO they can make is actually down. And so you may see anemias in chronic kidney disease patients. How this is managed is largely with exogenous erythropoietin. There are drugs that are similar to erythropoietin that are used. And finally, we get to hemolytic anemias. So previously, we've talked about a couple of hemolytic anemias. I never used the term hemolytic, blood lysing or splitting. So you're making the red blood cells, but they're getting damaged somehow and they're getting split open. Could be because of a genetic disorder like sickle cell or thalassemia. Could be because of immune system activity, inappropriate immune system activity. Immune thrombocytopenia purpura comes to mind. Mechanical hemolysis or hemolysis, prosthetic heart valves. They're rough on the red blood cells. If something comes on precipitously, so the most obvious sign is you have this big increase in unconjugated bilirubin, which I'll explain in a moment. What the patient symptoms are, they appear yellowish, their skin is yellowish, and their urine is dark. It's covered on these pages. Where does the bilirubin come from? This is covered in our textbook on these pages. This is the diagram. I'll walk you through. So, red blood cells, they don't last forever. 90, 120 days, then take them out of circulation. And the spleen largely, we recycle the parts of them, specifically the heme gets broken up and we take the iron out of the heme group and recycle the iron. The rest of the heme group is unrecyclable. And so it's trash. That ends up getting into the liver, is bilirubin. It gets conjugated, put together with something else, and then that comes out in the bile and it goes into the stool. And so that's how the body gets rid of most of this heme group. I'm going to show you the same information in a slightly different diagram because it adds a little bit more information. So as we've just said, red blood cells get recycled. You take the amino acids of the globin, the iron of the heme. Okay, so red blood cell here, we break it, take the hemoglobin, split it apart. The globin part, that's the protein part, we take all those amino acids, reuse those. The heme part, we separate that. We're going to take that iron, that is precious. We're going to keep that. It's the rest of that heme group. I don't need that and I can't recycle it. It doesn't matter. I was going to make a joke, but I'll skip that. Unconjugated bilirubin gets into the bloodstream. In the bloodstream, it eventually finds its way through the liver and the liver conjugates it and then gets rid of it here through the bile. And the bile duct goes to the small intestine and then gets pooped out. Some of it stays in the blood and can get peed out. So if you suddenly have a big uptick in how many red blood cells are getting split open, the liver gets overwhelmed, can't keep up with all the conjugating that it has to do. And so then we see more of this bilirubin in the bloodstream. Some of it gets deposited in the skin. Some of it gets excreted through the kidneys, and that's where the dark urine comes from. Is that color? Remember, the heme is a lot of the color of blood, and so that's getting peed out. And so the patient will see dark urine. What we will see, the patient may notice this too, again, it's this unconjugated bilirubin. When it gets deposited in the skin, it leads to a yellowing of the skin known as jaundice. If it gets deposited in the sclera of the eyes, and this is a good place to look too, because no matter how much melanin somebody has in their skin, you might not detect jaundice in somebody who has more melanin in their skin. All of us have whites of our eyes, and the bilirubin is quite obvious in the sclera. Technically, this is known as icterus. So here's a big summary of all the different causes of anemia. in my head, I just did this to organize things in my own head. So split it into loss of red blood cells or a problem replacing the red blood cells. So loss of red blood cells could be due to bleeding, they're hemorrhaging, frank blood loss, or occult blood loss in the stool. Could be due to hemolysis, the hemolytic anemias, congenital like sickle cell disease, or acquired if there's suddenly an immune system attacking the red blood cells. That's a hemolytic anemia. So either of those is going to decrease the amount of oxygen that the blood can carry. And the other problem may be I can't make red blood cells fast enough to replace them. It could be because the signal's missing, the erythropoietin, deficiency of erythropoietin due to kidney damage. Could be reduced ability to make hemoglobin because of iron deficiency or a genetic disorder. Could be because of a slowed DNA replication in the cell division due to a vitamin B12 or a folate deficiency. And last one, I didn't talk about this during the lecture, but there may be altogether an inability to make blood stem cells. This is a very serious condition known as aplastic anemia. Aplastic, so not producing an anemia. And this one's more complicated than just anemia. Yeah, you're not going to have enough red blood cells, but you're also not going to have enough white blood cells or platelets either with aplastic anemia. Okay, so there's the summary, and that is the end of part 1. See you in part 2.